Cell potency is a cell's ability to differentiate into other cell types. The more cell types a cell can differentiate into, the greater its potency. Potency is also described as the gene activation potential within a cell, which, like a continuum, begins with totipotency to designate a cell with the most differentiation potential, pluripotency, multipotency, oligopotency, and finally unipotency. Totipotency is the ability of a single cell to divide and produce all of the differentiated cells in an organism. Spores and zygotes are examples of totipotent cells. In the spectrum of cell potency, totipotency represents the cell with the greatest differentiation potential, being able to differentiate into any embryonic cell, as well as any extrembryonic cell. In contrast, pluripotent cells can only differentiate into embryonic cells. A fully differentiated cell can return to a state of totipotency. The conversion to totipotency is complex and not fully understood. In 2011, research revealed that cells may differentiate not into a fully totipotent cell, but instead into a complex cellular variation of totipotency. Stem cells resembling totipotent blastomeres from two-cell stage embryos can arise spontaneously in mouse embryonic stem cell cultures and also can be induced to arise more frequently in vitro through downregulation of the chromatin assembly activity of CF1. The human development model can be used to describe how totipotent cells arise. Human development begins when a sperm fertilizes an egg and the resulting fertilized egg creates a single totipotent cell, a zygote. In the first hours after fertilization, this zygote divides into identical totipotent cells, which can later develop into any of the 3 germ layers of a human, or into cells of the placenta. After reaching a 16-cell stage, the totipotent cells of the morula will differentiate into cells that will eventually become either the blastocyst's inner cell mass or the outer trophoblast. Approximately 4 days after fertilization and after several cycles of cell division, these totipotent cells begin to specialize. The inner cell mass, the source of embryonic stem cells, becomes pluripotent. Research on C. Norabditis elegans suggests that multiple mechanisms including RNA regulation may play a role in maintaining totipotency at different stages of development in some species. Work with zebrafish and mammals suggests a further interplay between miRNA and RNA binding proteins in determining development differences.